Hello, everyone. Okay, can you hear me well?
Yes.
Yeah, good, thank you. Well, hello, everyone. And well, first of all, thanks the organizing committee for inviting me to give this talk. I work at CREAN, a Center for the Study of Harmful Algal Blooms in southern Chile, which is part of the Chilean Fisheries Development Institute, IFOP. Well, my talk is entitled Understanding Impacts of HABs on Fish Farm based on lessons learned in Chile. Next, please. Well, for those who haven't had the chance to visit this country, Chile extends from 18 to about 56 south latitude and is about 4,000 kilometers long. In terms of marine currents, you can notice on the upper right corner that South America is the only landmass to interrupt the subtropical front. Next, please. So the west wind drift defines the Humboldt Current to the north with intense upwellings and the Cape Horn Current to the south. Next, please. In general, this complex interaction also determines the northern extent of the South Antarctic surface water, which represents a severe deficiency in silicic acid, potentially playing an important role in the niche competition between these two big groups, the diatoms and the toxic flagellates. So then you can notice that the southern Chilean coast is in a unique oceanographic position, not only influenced by the presence of South Antarctic surface water, but also by the regular input of fresh water from ice melt of glaciers. Next, please. Well, and these particular oceanographic conditions in the south of Chile have promoted the strong development of the shellfish and the salmon farming industries, as you can see in this picture that shows the socioeconomic activities along the Chilean coast. Next, please. The salmon industry started in 1982 and now produces around 25% of the world supply. Next, please. The main culture species is the Atlantic salmon, but there is also production of coho salmon and rainbow trout. Next, please. The production is mainly located in the Los Lagos and Aysén region and is expanding further south into the Magellan region with more than 400 fish farms in operation. Next, please. Well, related to harmful algal blooms in Chile, we have some world records that have been described in literature. For example, next please. Some of them related to blooms that have affected human health, blooms that are associated with toxins, but from now we're gonna call them true phycotoxins to differentiate them from ichthyotoxins. In 1970, it was reported the first intoxication in the world with DSTs, these diuretic shellfish toxins, with more than 200 people intoxicated due to Dinophysis acuta. And then two outbreaks in 2018, one of Alexandrium catenella that produced the highest worldwide PST concentration ever recorded, with more than 140,000 micrograms of saxitoxins in 100 grams of product. We have to notice that the regulatory limit is only 80 micrograms, and a high concentration of Dinophysis acuta, around 120 cells per mil in a fjord in the south of Chile. And next, please. Related to fish-killing algal blooms, a very known bloom produced by the Dichocophytes chatonella that produced enormous losses to the Chilean salmon industry in 2016. Next, please. Well, related to research in Chile, most of the efforts in the last decades have been focused on true phycotoxin producers, such as the PST producer Alexandrium catenella, for its strong impact on human health and aquaculture since 1972 in Chile. Next, please. So then we know a little about ichthyotoxic species, such as the flagellate Chatonella, the Rhaphiophyte Heterostigma, and the Careniacea family. Next, please. It is important to highlight that microalgae species that produce true phycotoxins, I mean, paralytic, diuretic, and amnesic shellfish toxins are not the same species reported to produce fish kills to gill damage, or at least we are not talking about the same toxins. However, you can notice Alexandrium catenella at the bottom of the pie chart produce both PSTs and ichthyotoxins. Next, please. After some research on Chilean strains of Alexandrium catenella, these two studies conclude, next please, that paralytic shellfish toxins do not produce fish gill damage, but rather this damage is in part actually attributed to the synergistic reaction between reactive oxygen species and polyunsaturated fatty acids, well, and also probably other unknown toxin metabolites. Another outcome from these studies is that ichthyotoxic activity may increase after cell rupture, which is important when using mitigation strategies that could destroy the fragile toxic flagellates and then release all these toxins into the water. Next, please. Well, now in the next slides, I will briefly show you some of the hypotheses of the causes of why we are observing more intense HABs in the Chilean fjords in the last years, related to climate change, the flushing capacity of the fjords, and potential eutrophication. Next, please. Examples of socioeconomic impacts and effects on the ecosystem. Next, please. And some observations about mitigation strategies applied during some blooms, monitoring and analytical methods for ichthyotoxin detection. Next, please. Well, related to climate change, we observed that a massive outbreak of Pseudochatonella in Alexandrium in 2016, next please, coincided with the driest summer in the last 70 years in Patagonia. Next, please. And the same trend was observed during the second driest summer last year with an intense bloom of the rhaphdophyte Heterosigma cashew. Next, please. So now related to the possible causes, so here at the bottom right, there is an image showing the complexity of the coastal currents in northern Patagonia. So basically a slight shift in these currents are going to have huge impacts. And what in fact was seen in the two events that I showed you before was that, next please, the west wind drift current did shift south to the position of the high pressure in the Southeast Pacific during these particular years. So this condition caused positive temperature anomalies with very dry summer-fall seasons. Next, please. This article, published in Climate and Atmospheric Science, shows in the magenta line modeling of the subtropical ridge of the Chilean coast from 1970 to 1990, I think. And the green line shows the future position of the subtropical ridge. So we can see that there is a slight southward movement of this ridge that is going to continue in the future. Next, please. An important effect of less freshwater input during these dry periods is the reduced flushing capacity of the fjords. And you can see in this simulation, Ross, if you can press play on the left graph. No, the previous one. That you can press play there on the graph. Yeah, there. Probably it's not working. Well, the particles were moving there in the fjords, and they got retained into the fjord during the outbreak of Heterosigma. Well, the next one. But you can see in this picture from that event that water retention can enhance cells' patchiness and also keep ichthyotoxins in the water for longer. Next, please. Well, this picture shows the high fish mortality during that event, more than 6 tons. Next, please. Based on the flushing capacity of the fjords in northern Patagonia, and this paper published last year estimated the eutrophication risk for this area. So here, darker red means a higher risk of eutrophication. Next, please. Interestingly, these fjords are precisely the ones that have presented the most intense blooms in recent years. Next, please. Some of the impacts on the ecosystem are the high biomass blooms that we have observed more frequently in recent years in the fjords. One common species now is the dinoflagellate Lepidodium chloroforum. And in this year, we had this massive bloom of Tetraselmis in the bottom middle panel. And in this right moment, in front of my house, actually, we have this massive bloom of Proocentrum micans. Next, please. And then next one. Then it should start the video. Probably we cannot play the videos. Yeah, but well, it was a very interesting video. That well, other impacts in the ecosystem are the cytotoxic species that affect wild marine fauna, such as this bloom of Carenia and Carlodinium in 2017 affecting sea urchins. Next, please. Related to the socioeconomic impacts, we have this very known bloom event in 2016 called the Godzilla Red Tide event, with first a massive bloom of Pseudochatonella and then an offshore bloom of Alexandrium catenella. Next, please. The Pseudochatonella event produced losses to the Chilean salmon industry for about $800 million US, and it's considered the world's largest farmed fish kill caused by a harmful alga bloom so far. Next, please. During this outbreak, the mitigation strategies used at the salmon farms were not successful. Next, please. Then the next, yeah, probably because of the most used technique were the upwelling system and bubble curtains that pumped water from dense Pseudochatonella thin layers up to the surface, making the thing worse. Next, please. Currently, our group is working on answering some questions about the efficiency of these methods. For example, if curtains can really block the horizontal transport of cells into the salmon cages. And next, please. If these systems destroy the cells, releasing the toxic intracellular content into the water. Next. Well, there are important observations about monitoring. Fish killing flagellates usually have complex life cycles. They are pleomorphic, so they can change their shape and are very fragile. So they do not resist fixatives such as Lugol or formalin, which make difficult to monitor them using light microscopy. So here in these pictures, you have examples of how these cells look when using fixatives. Next, please. So then new technologies needed for this type of microalgae, for example, molecular approaches and or automated flow cytometry that allows analysis of light samples. And probably Alejandro will talk about that later. Next, please. In 2018, we hosted in Puerto Varas in the south of Chile a workshop to talk about the fish killing marine algae and their effects. And one of the key points was the lack of an analytical methods to detect ectotoxins. Next, please. So as we use the mouse stays to detect, for example, the paralytic shellfish toxins. Next. It's been proven that the RT-gill cell line from rainbow trout can detect ectotoxins in the seawater. Next, please. So based on the principle that ectotoxins attack the cell membrane of the gills. Next, please. Then by culturing gill cells, it is possible to quantify the cytotoxic potency of fish-killing algae from natural water samples. Next, please. So currently, we are working on improving this assay in our laboratory for using it in regular monitoring of ectotoxins. So this picture shows how the biocell looks like. Next, please. Well, and finally, some take-home messages. Well, the use of existing HABs mitigation strategies currently used at salmon farms need to be reassessed based on new studies. Regular monitoring of fish-killing algae needs to be accompanied with new technologies to improve detection resolution. It is necessary to standardize a common method for the detection of phytotoxins, for example, the GILS assay. And finally, climate change is strongly affecting the southern Patagonian fjords in recent years and is adding much uncertainty about future HABs events. Thanks for your attention, and I'm open for questions.
Ross, you gotta unmute your microphone.
Thank you very much, Jorge. That was an amazing talk. So I'd just like to invite people to ask a couple of questions. If you put your question in the chat and then put it to Jorge. I don't know, Mufida, do you want to ask a little bit more about the molecular tools that Jorge was talking about? Would you want to switch your camera on and switch your mic on?
Well, basically, well, some of the tools that we have at the laboratory now are trying to put in the monitoring system in Chile. They are quite expensive. That's the first problem, but we are using metabarcoding. So then you can have a great amount of genetic material, and you can identify a lot of species from the water. Some species that also you cannot identify using the light microscopy. So small flagellates, and that basically can get destroyed after using fixatives, so you cannot identify them. So in this case, the metabarcoding analysis can give you a big picture of what is the phytoplankton or microalgae assemblage that you have in the water. And also you can use qPCR to quantify species. They have some limitations because you have to develop a lot of tools before to establish this kind of analytical tools in the laboratory. But it's important to understand that monitoring algae that is affecting the human health, for example, and these dinoflagellates or diatoms, so they are easier to identify at the microscope. But all the flagellate, the small flagellates that are toxic, they need more tools to have a more clear and fine resolution for identification and quantification.
Hello, George. You do culture in your lab? You isolate the species?
Yeah. Well, our laboratory is quite new. We started working in 2017, but at the moment we have a nice amount of different cultures.
You have a culture collection. You have a culture collection.
Yeah, and basically there we do a lot of experiments, trying to understand well the responses of these species against different drivers, environmental drivers.
Yes, thank you.
Okay, thank you. One more question. I think we have a question here from Alam about do you use rapid tests for biotoxins in Chile?
Rapid test. Well, there are these tests that you can use for checking if you have, for example, parasitic toxins. But we are not using it now in Chile. We tried before, but basically because we have to fit this rapid test for to match the toxin profile that we have in our strains in Chile. So you can have Alexandrian catenary in different regions of the world, but they have different profiles. So that you have to fix a little bit these tests for having something successful with your own strains. But at the moment, we are not using them.
Okay, thank you very much, Jorge, for presenting today. That was amazing. Thank you. If there are any more questions and you want to follow up with Jorge, then we can arrange that after the workshop. So I'd like to move things on, ladies and gents. The next speaker will be Dr. Isaac Yongguan Huan. And it's really great that you can join us today, Isaac. So you're a professor at the Institute for the Oceanology for the Chinese Academy of Sciences. And your research focuses on eutrophication and the control of harmful algal blooms. Mm-hmm. So very grateful for you to present today, and we're just going to hand over to you.
Yeah, can I just share my screen now?
Yes, please go ahead.
Yeah, but you know, from the Zoom, it is shows that I could not use this.
You should be able to share it just now. Try again.
Okay. Yeah, it works.
Okay.
So is it clear? Very
clear.
Okay, so let's go. So hello everybody, I am Isaac from Institute of Oceanology, CAS, and my own interest of my research is about the coastal eutrophication. You know, the nutrients and things, where does it from? How many should be input in a typical area, and is there any relationship with the coastal eutrophication and the HABs? But today I will share a story about the control of these half-algal blooms in China. That we used a technology called modified clay, which is initially proposed by our PI, Professor Zheng Yu, who is the man at the center of this picture, wearing an orange shirt. So let's start now. As we all know, that the HABs is a global ecological disaster. Especially, it has many negative effects in the RSA area, in Miromis areas, and it has the negative impacts to the human health and to the agriculture, and maybe for, I don't know, for the desalination power plant. So let's talk about the HAB management approaches. Well, it could be categorized into three categories. Initially, the prevention. It means we could do something before the HABs just outbreak. We could do some daily routine, something like that. And the second category is called the mitigation. We could find some method to reduce the losses of such HAB disasters and to the economy or to the environment. The last but not the least, the third category we call the control, to eliminate or suppress the HAB cells just in field. So there are several methods about prevention and mitigation of the HABs in the agriculture areas, and there are several several methods just listed here. We could find a good site for the agriculture and to conduct some daily routine monitorings. And when the HAB just outbreak, we could stop feeding the fish or other cultural organisms. All of these actions will help, but you know, it will not stop the HAB immediately. We always say that the HAB disaster is just like a fire. It could outbreak suddenly. It could just influence a very large scale area, and it has very negative effects to the humans. So for the fire, we should have something in hand. And for the HAB, we also should get some control method in emergency, just like a fire extinguisher. And for the emergency control, there were several methods have been, just studied in the lab scale, but few of them could be applied in field. For example, for the physical measures like ultrasonic or something else, it could be useful in the lab scale, but maybe when we just face maybe 100 or 1,000 square kilometers types in field, it is could not be used in field, right? And for the chemicals, we used to apply spray the copper or anthracite onto the HAB waters several years ago. But this kind of method was prohibited right now because of the secondary pollution. And it's biological measures, which seems very promising, but it's just in the lab scale research, it could not be used in field. Besides this method, the flocculating control, which maybe the only method could be used in field by now. So in principle, and the clay particles or could just form some just could be flocculated with the HAB cells and to become some little some small flocks, and the small flocks together become the big flocks and set down to the bottom. That is the principle that the flocculation control could be used for. So why should we use clay? Because we could find it anywhere. It could be anywhere from the natural soil. It's abundant. It's relatively inexpensive. It also could be found in most aquatic sediments, which means it's very green and natural, material. It has low environmental impacts. And last but not the least, it has been used successfully against the HAB disaster in several countries. So that is a case which I show here in the South Korea. And they used the yellow soil, yellow clay, and to be a routine or to be a routine HAB against approach in their countries. They tried several methods, but now they find that the clay spray may be only the promising approach that could be used in field. And that is the pictures which I showed here is how they just spray the clay onto the HAB cells. And this method has just been used in Japan and in the US. And I found very, very interesting, the Iran has just sprayed the clays onto the HAB cells either. They use the airplane. Well, it's very interesting. So now what we did in China, we used the modified clay, a kind of clay, and we used this kind of technologies in open waters to face 100 square kilometers HABs. And we also use the MCs in the aquaculture size. The left panel is what we did in the shrimp culture ponds, and the right one we used, to use in the abalone culture size. And we also use this against the HAB disasters in the new nuclear power plants. Yeah, here is an example. And in these areas, a HAB dominated by the Phyllocystis globosa just occurred. The globosa could get some big colonies, and massive colonies could just proliferate very massive and could clog the filter and so that maybe get some problem for the cooling water intake. So we use our methods against such HAB disasters in this area. Maybe there is some similar stories in the desalination power plants in the RSA area. I don't know. So why this technology could control HABs? First, it could just flocculated with the HAB cells effectively. We call that the directed function. By these functions, maybe 70 or 80% of the HAB cells could just be settled down to the bottom in a very short time. But there was only maybe 20% or 30% HAB cells could not just be eliminated from the water by these directed functions. And such kind of cells, the density of such residue cells should be very, very high. And what is the fate of such residue cells? We found that they could just not grow up and could not germinate by itself. And such proofs were just tested in the lab, and it is found that the residue cells experience the physical biochemical stresses because of the MC addition. So we just concluded the mechanism of MC controlling HABs into two parts: the direction functions, and which just happens in the water very quickly, and to settle down the HAB cells maybe 30 minutes to 1 hour. And we also have the indirect functions, which means because of the MC additions, the residue cells, the growth of the residue cells would be inhibited. And that is why the MC could just control HABs in field. And the efficiency of this modified clay is much higher than the natural clay. The dosage we used is only 4 to 10 tons per square kilometers, which is very limited comparing to the natural clay. So the reason why is if we just to look at the interactions between the clay particles and the HAB cells, and these natural clay particles, the surface charge of these particles is negative, and the surface charge of the HAB cells is also negative. So the negative with negative, the interaction between these two kind of particles are repulsive. So we used some artificial methods and to modify the surface charge of the clay particles from negative to positive, and by adding some the polymer, the long-chain polymers, and to increase the positive charges of the clay particles. So the interactions between these two kind of the particles from repulsive to attractive. So that is the reason why it is very much efficient than the natural clay. So that is a proof from the global bloom controls in the NGP area. So when the colony's density is high, we use the MC and spray the MC onto the HAB waters, and the cell density just decreased. So it's a very environmental friendly technology either for the MC. It could just absorb the nutrients in the water and to improve the water qualities. It has no side effects for typical aquaculture organisms. And it could absorb toxins either, and it could inhibit the germination of the seeds of some typical HAB species, and it could improve the biodiversity for the ecosystems. And this method was evaluated by several third parties, and it proved to be no harm to other organisms, and we would favor the environment. And that is this table shows we have experienced several experiments of this method to typical aquaculture organisms like shrimps, brownie, and scallop, sea cucumber, and others. And that is what we tested any potential of this, any potential effects of the MC and to the salmon, to the Atlantic salmon. So on the right video, we just spray the MC much, maybe 10 times of the dosage we used in field, and we've tried to figure out is there any potential effects to these salmons, and the answer is no. The respiration rates and the swimming abilities between these two groups are the same thing, and there is no cuff for the MC addition group. No fish died in this MC spray group, but we find one died fish in the no MC group, maybe because in the MC addition group and the water quality just improved. I don't know. And so no visible differences of the gels were found on the microscope review. It reveals that the MC didn't affect the fish gills, even at a much higher loading over the dosage. And we also used this method in the shrimp culture ponds, and like this video shows. And it is is very amazing that the growth of the shrimp is just much faster than the control growth. Maybe because by use the MC, the community structures has been just adjusted, and the HABs, the potential risk of the HABs just be avoided. I don't know. So
are we coming to the conclusion?
Okay.
Thank you.
Yeah, it is very easy to use, and we just studied several equipments, and that could be used in the culture ponds and in the open waters. And actually, it has been used in the over 20 waters along China coast. It's a very promising technologies to mitigate the HABs in the world. And it has been just introduced to other countries. Here is it's just a case we collaborating with the Mons laboratory and Professor Anderson, and in the US. And it also has been introduced to Peru and for Chile. Actually, in the 2016 Chile delegations, including the officials, experts, and managers, come to China and to view these MC technologies. And we have some MOU here, and have some meetings with the governments, and have some workshops. And such collaborations was just reported by Chilean press. Okay, so the final source, so the many HABs are disasters and should be treated as such. So the public and authorities and also the aquaculture industries want some kind of the HAB control. So there are many some promising strategies that could be used in field. One of which is the clay disposal. So in this field, MC is widely used for the bloom control in China, and has the potential to be used in the RSA area. So that is my take-home message. And thank you for your listening, and I appreciate your questions.
Thank you very much,
Thank
Isaac. If you could stop sharing your screen, please.
Yeah.
That would be great.
Okay.
Thank you. I noticed there was one good question in the chat already from Claudia. Would you like to ask your question to Isaac?
Yeah, sure. Thank you, Isaac, for your presentation. The question is a basic question that I'm going to read, actually. Knowing that the Arabic Gulf and especially the Red Sea are one of the most pristine coral reef, do you think that clay can be applied in this environment without having impact on the coral reef?
Yeah, it's a good question. And by now, we actually didn't do the experiments with the coral reef because we didn't have coral reef in Qingdao, We could test is there any potential effects to the coral reef. But in my opinion, I don't think it has some negative impacts to them. It could just improve the water qualities by MC additions. It could absorb the nutrients, and it has no side effects to the typical aquaculture organism like shrimps, like salmons, fishes, abalones, sea cucumbers, and several organisms. I don't find the reason why it could have negative effects to the coral reef.
Okay, thank you very much.
Okay, thank you.
I'm afraid we're going to have to move on, but if there are any more questions for Isaac, we can follow up on those after the workshop. I'd like to move on, please. So it's my pleasure to introduce our next speaker and to take a shift back to the RSA. So this is the Rokmi Sea area, including the Arabian Gulf and also the Sea of Oman. So first of all, we have Dr. Qusai Karam. He's my co-host for this workshop. Qusai has 19 years of research experience as an aquatic toxicologist. He's also a marine geochemist and has done extensive research in the field of aquatic toxicology. and the effects in particular of petroleum hydrocarbons and oil spill dispersants. He's employed multi-species tests, including marine algae, invertebrates, and fish. So yeah, without further ado, over to you, Qsai.
Thank you very much. And good afternoon, everybody, or good morning or good evening again, depending on where you are living now. So my talk today is the development of an early warning system for harmful algal blooms, red tides, and fish kill incidents in Kuwait territorial waters. So the whole objective of the talk is to develop an early warning system based on the fish kill incidents that occur in Kuwait. Now, in Kuwait, we have multiple sources of pollution, point sources of pollution such as sewage outlets for untreated sewage that create a load on the marine ecosystem, thus the nutrients load in the aqueous water column. And we have reported the elevated levels of nutrients in the wastewater, especially with increasing water temperature in the summer, and the salinity can trigger harmful algal blooms and algal blooms, especially in the marine waters, which can be a precursor for subsequent red tides and fish kill incidents. Also, reported incidents of harmful algal blooms, species responsible for selected marine mortalities in Kuwait, such as different fish species, as we will see in the this presentation here, lay the ground for and the foundation for developing an early warning system to forecast and predict such environmental cases. So the Kuwait Environment Public Authority has requested from KISR, from the Crisis Management and Decision Support Program, to submit a research proposal to assess those marine crises and to develop kind of an early warning system for forecasting and predicting, and later by the decision makers to mitigate those issues. Consequently, the idea, as I have said, merged into the integrated system of prediction and forecasting. We call it the early warning system. It will incorporate various analytical tools that what is available at KISR and Kuwait EPA in a joint operation room between the two, in addition to the ROBME organization at Kuwait and in the Gulf, to analyze HAB, red tide, and fish kill events. On all these photos show the fish kill incidents in Kuwait here, with the red algae and red tide here in Kuwait every summer, but in different magnitudes. So normally we use remote sensing to visualize, to see the algal patches from space, and as the first step for the early warning system to visualize and to see and to assess those algae in the Kuwaiti waters here, as been demonstrated here by these photos provided by Professor Petrov. Another one here showing the algae accumulating here in the Kuwaiti waters. That will serve as the first stage of the early warning system. So these reports been generated in KISR in our program, the Crisis Decision Support Program. They have been handed over to EPA to identify the stations or the locations where the algae accumulating in the Kuwaiti territorial waters. That is this area here. Reporting for chlorophyll concentrations in the Kuwaiti territorial waters. And these reports been generated over the years since we developed the remote sensing unit and our program here. It's been a great addition to the team. And this is clearly demonstrate the concentration milligram per cubic meter of chlorophyll per the as the report have detected. We have a major issue that links the HABs, red tides, and fish kills. That is as I have said, the discharge of treated and untreated sewage here in Kuwait. Doesn't mean that it's a treated sewage that doesn't create an issue. It's because of the nutrient loads that the algae kind of feed on and act as a nourishment for those algae. They will flourish further and bloom, thus causing and devastating the marine environment and causing the fish kill incidents that we have reported in 1999 and 2001. That was the major HAB incidents and fish kill incidents affecting the Liza Macrolopus species, that is the mate, locally named mate. It's a mullet, which is of an economic value to the country and the region. And we have a lot of, in 1999, 25 to 30 tons of mullet fish been devastated and were lost. And in 2001, another major fish kill, nearly 2,500 tons of mullet fish were lost to the red tide and the algal bloom incidents. And this photo demonstrates the dead fish, and this is the decaying of the algae being cornered here next to the desalination plant. It's a small embayment near the Shuwaikh area, where the decaying algae were driven by the water current, and further they decay further here in this region to lead the further decomposition. I have a video here, but I don't know whether it's going to work or not. Same issue. It shows the fish kill in Kuwait. Maybe I can play it later. So this map and the next one kind of demonstrates all the sewage outlets in Kuwait Bay and Kuwait territorial waters. This is part of a project that we conducted with Dr. Yusuf Al-Asiri. He presented his work yesterday. We kind of surveyed all the sewage outlets, wastewater outlets here in Kuwait, with a total of 78, approximately 78, 80 outlets all over Kuwait, mostly located in 30 of them located in Kuwait Bay, that would act as a stressor to the marine environment. Now, some of these are permanently closed. Some of these are 24/7 discharging wastewater to the Kuwaiti marine, affecting the ecosystem itself. And thus the other ones are, properly discharging waters. So these stations by Kuwait EPA, 12 stations, have been distributed all over the Kuwaiti waters, most specifically in the Kuwait Bay, which is a hotspot for red tides and issues because of all the sewage outlets discharging wastewater here, treated and untreated. So we have multiple of these and all over. And we use the data and the future research and current research to classify and assess all the water quality parameters, as the nutrients, petroleum hydrocarbon, trace metals, and the changes in water quality that will assess our research and future research into the early warning system. So as a second part of this presentation, what I have done over the years is to collect all historical observation, changes in water quality, and incidents of harmful algal blooms, including the species, and the fish kill incidents, and try to organize them in a kind of a table form to see what are the main triggers or main changers or players in those three major incidents: the HAB, the fish kill, and the red tides. They are all correlated to each other. So I have developed kind of a manuscript that I would like to share with you in the future, that you can have an input on it, so we can assess the status of the environment, particularly to the harmful algal blooms and the fish kill incidents. That will cover almost 20 years since the 1999, the major fish kill, and the species, the causative agents of those fish kills, and the amount of chlorophyll till the recent one up to the COVID and post-COVID period. And those species been reported by our colleague, Dr. Faisal Imani and Pauly Karbov in 2012, highlighting all the most harmful algal species in Kuwait territorial waters described in this paper here. So we come back to the early warning system that we have tested in Qusr using our multi-tool system. The first stage of this early warning system, it's been tested successfully, is to monitor the HAB or the algal patch using a remote sensing techniques and satellites, the Aquamodus satellite with the 250 resolution, 250 meters resolution. The report you have seen in the presentation will be sent to the EPA team to collect sample, and those data will be communicated further to the numerical modeling team, Dr. Yusuf, who spoke yesterday, who will use his physical parameter data, the water quality, wind, and current, to generate a model, a numerical model, to predict where the algal patch will travel further. Well, it will impact recreational facilities, desalination plants, according to the current wind speed and tidal movement in Kuwait, particularly in the season where the HAB occurs. That is mostly in the summer, but you get a different atmospheric conditions every year with the climate change and so forth and so on. So once we identify remotely from space where the most algal patches kind of congregate and accumulate, the biology team, the sampling team from EPA, our collaborators, will collect samples and classify those samples as harmful or not harmful, and what their concentrations and so forth and so on. So using all this multi-layer early warning system, we can incorporate all the data that we have into the GIS system to generate hotspot maps and further analytical maps that demonstrate where most species been collected, what are the seasons, what are the hotspot localities that the harmful algal blooms been targeted in the Kuwait marine waters and all the territorial waters. So this is briefly describes the techniques or the tools that we use. For example, in the monitoring system, we use a spectral reflections of plankton species from space to identify the algal or chlorophyll concentrations using the modus aqua, as I have mentioned to you. In the numerical modeling, we use the Delpit 3D for field measurements, for incorporating all the water quality parameters, salinity, water level, temperature, and dissolved oxygen, and all rheological aspects will be evaluated using a model and a flow model. The biology team at EPA will collect seawater sample, fish samples to identify most affected and classify them. And then we layer them over into GIS team, which is another team at KISR, and the same operating room for this early warning system. And this is all the steps that highlight the early warning system tested in Kuwait. And we have a newly accepted project for funding by Kuwait EPA to establish this early warning system between KISR and EPA. We have tested the system, it's successful, and it's a versatile and very interactive, and we are willing to introduce a new topic, new segments to this early warning system, specifically toxicology and genetics for next generation sequencing. And this is the data acquisition, another kind of graph demonstrating the forecasting and the early warning for decision makers to respond to it. So it will be a tool for all decision makers and authorities and regulatory authorities in Kuwait here to issue a warning in case of a spread of a HAB incident, to not swim in the recreational facilities, to not drink or not fish of any harmful algal species. So, and this is the buoy that our colleague Dr. Yusuf used to collect water quality and incorporate it into his mathematical model here. We call it KISR 01, owned by KISR to document all changes in the water quality. So key discussion points and take-home messages. What I have observed and assessed from all the researches that have been conducted that I utilized to develop an early warning system and future research for HAB and red tide and fish kill, that HAB incidents in Kuwaiti waters consist of toxic and non-toxic species as per Kuwait EPA reports. The reoccurrence of specific neurotoxin HAB species over the years have been recorded in Kuwaiti waters, which is of a major concern due to the status of the fish or the moribund state of the fish we have seen when we collect sample, and specifically in 2001. Emergence of new species in the Rob Sea areas that have been documented and elsewhere, specifically in the Sea of Oman, just like the Blacocortius species. Now HAB events frequencies are elevated in the summer season. So we have major events in 1999, 2001. Then over the years we have a small incidents. Then later we have a not as major as the 2001, but it's a reoccurring concern and worry. And we have identified too that Kuwait Bay and the North Arabian Gulf, which is the end of the spectrum opposite to the Sea of Oman is a hotspot of HAB and red tide because of the sewage outlets and discharging here. The wastewater is confirmed in literature and with our colleagues from CFS, their research, that is the causative for HAB and red tide. And thank you.
Thank you very much, Kuzai. That was a really great talk.
Thank you.
So I'd just like to offer the opportunity for the audience to ask a couple of questions. Can you pop them in the chat? Or if you feel bold, just ask your question directly to Kuzai.
Hello, Dr. Ross.
Hello, please go ahead and ask your question.
Hello. Thanks, Doctor, for your informative introduction about early warning system. My question is not a question, it's a suggestion, because from this workshop, we know there are many problems we have in the Gulf, but not much data is in the HAB system. Dr. Ross introduced yesterday. So in the last year, we published the global harmful algal status report. So that region is not analyzed. So I would suggest maybe if you have interest, you can put your data there, because just now I saw a slide. There are several harmful events there, but I just checked the system. There's only one event in our system. So it would be good if these are included into this platform. So for the next report, we can include that for the global analysis.
That'll be good, actually. That'll be good.
Yeah.
I mean, this is the part of the whole workshop is to knowledge sharing and data sharing, that will be aware of existing platforms that we weren't aware of, and we thank you for it, and the participants that we are willing to share whatever available, because it's your expert opinion that we will benefit from in analyzing all these data. Thank you. We'll put that one in the schedule, and we'll see to it immediately. Thank you, Doctor.
Thank you.
Thank you very much, Jan. Thank you. Okay. Any more questions? We've got time for one more question if someone would like to ask Kuzai, and then we'll move on. Okay, well, if anything occurs, again, we can follow up after the workshop. Thank you again, Kuzai. I'll let you get back to your other job.
All right, I'm gonna switch hats now. So
okay.
Next, I'm just gonna select, yeah.
Yeah, thank you. And it's with great pleasure that I'm going to introduce our next speaker while Kuzai gets the slides ready. So next we have Dr. Gilan Ataran Fariman. Gilan is an associate professor of marine biology at Shabahar Maritime University in Iran. She conducts research on HABs in the Eastern Arabian Gulf and the Sea of Oman. She advises the Iranian Fisheries Research Institute on HAB risks. So, Gilan, could you please switch your camera on and take the floor? The floor is all yours.
Hi, everybody. Is it okay?
Yeah,
Yes.
I'm trying to share, but there is a lag in the system, so.
We can see you, we can hear you, Gilan, so that's all good.
Okay. My presentation is about existing HAB observation and early warning system in the Persian Gulf and the Sea of Oman. Next, please.
Because I have two presentations open here, so let me just work on it a little bit.
Can I change my slide myself?
No, I'm going to change it for you.
Okay.
So
Next,
is it fine now?
Yeah.
Okay, great.
Okay, next, please. The main source inducing HABs, no, sorry, previous one. The main source causing HABs are domestic, industrial, agriculture, discharge, agriculture, runoff, coastal and land sea filling, and ballast water. Next, please. The total amount of HAB species in the Sea of Oman and the Persian Gulf is only 6% of total phytoplankton in this area. And toxic species among HAB species are 36%. These data are based on this source and the numbers provided since 1979. Next, please. 14% of HAB species are diatom, and this table shows the common diatom HAB former in this area. Next, please. 43% of HAB former species belong to dinoflagellates, and these tables show the most common HAB former dinoflagellate in the area. And as you can see, Margalophydium and Goniolax and Carenia seriformis and Gyrodinium instratum are most common HAB former in this area. Next, please. 8% of HAB former belong to other groups of dinoflagellate, and species such as Chatonella and Heterosigma and Phaucistis among the HAB former contain only 8% of HAB species in this area. Next, please. 78% of mortality due to HAB caused by dinoflagellates in the Persian Gulf and Sea of Oman based on RAPMI, and 11% caused by diatoms, 5 and 6% caused by dinoflagellate and diatom and dinoflagellate and cyanophytes. Next, please. The key point is dinoflagellate are most important HAB former in the area, and most dinoflagellate HAB former species produce resting cysts during their life cycle. And the species in the water column can be estimated by cyst assessment. Next, please. Cysts act as a seed population. That means cysts as the seed bank play important role in the onset of plankton bloom that may happen, and therefore they can provide early warning for the presence of toxic species in a particular area. So study, next please. Due to catastrophic occasion in 2008 in the Persian Gulf and the Sea of Oman, many research projects have been conducted, and one of these have been carried out by myself with this title, Dispersion and Abundance Assessment of Different Species of Phytoplankton Cysts in the Sediments of the Oman Sea. This research project have been done in 2016, and in this project 47 stations in 16 areas during 4 seasons was sampled and studied. In this project we identify 83 species of dinocysts during 4 seasons. And do you have my voice?
Yes, we can hear you fine.
Oh, okay, sorry. And many HAB former species were identified in this project. Next, please. Many HAB former species such as Gymnidinium catenatum and Pyridinium bahamans that have been reported previously by other researchers, such as the study of Zandveld and et al. In 2013 showed the distribution of these 2 species from Arabian Gulf, Arabian Sea, and the Sea of Oman. And Margalophydium polycricoid for the first time in the sediment we found in the sediment in 2016, and this is Coclidinium or Margalophydium polycricoid from this area. For the first time, this cyst was with high abundance in the Strait of Hormuz was observed. Next, please. The next project was also carried out by myself with the title of Identification and Distribution of Phytoplankton cysts in the sediment from RAPME Winter 2006 Oceanographic Cruise, and granted by RAPME organization. And this project was done in 2020. And in this project, the sediment from 25 stations belong to 2006 was sampled, and that's been before catastrophic 2008. And the main goal of this project was assess the presence of Margariphidinium cyst in the sediment of 2006. Next, please. The result of this project showed that 99% of cysts belong to dinoflagellates, and as you can see in this map, the percentage of HAB cyst species to the others, and shows in the UAE, Oman, and Strait of Hormuz, the percentage of HAB species is higher than other species. And this chart shows that the main mortality in the Oman due to HAB was the highest amount in comparison with other countries, and the second one is Iran, and then Bahrain. This data is based on RAPME data in 1988 and 2000 until 2008. Next, please. This is Cochlidinium, I used to say Cochlidinium, Margariphidinium polycricoid cyst in this area. And as you can see in this map, shows the high abundance and high density of these species in the UAE and Strait of Hormuz station, this area. And this is the high abundance of Margariphidinium cyst in this area. Next, please. This is Pyridinium bahamans cyst, and this species is a bloom former and typical warm water species, and was previously dominant in the area and distributed in the tropical and subtropical region. And this map shows the distribution of this cyst, but when we compare with abundance of the Margariphidinium cyst, this species had very low concentration in the area. Again, UAE station had the highest cyst of these species. Next, please. All these sediments belong to 2006. From this point onward, I'm going to show you some of the bloom events that happened in the last few years, especially in the Sea of Oman. As you can see in this map, this is Goniolax polygramma. We observed bloom of this species in 2017 in the coastal water of the Oman Sea, Iranian side. First reported in 2008 and then 2006 from Arabian Sea and 2017 from Oman Sea. These bloom events cause water discoloration and many foam in the coast, and phylogenetically a study of this species showed that Goniolax polygramma from Oman Sea is closely related to the Korean strain, as you can see, with 100% similarity. Next, please. This is another event of macroalgae bloom in the coast, north part of Oman Sea. This coast is near the desalination plant station, and and cover large area, and lots of foam was there, and many microorganism, probably that zooplankton or other protozoa were dead in these bloom events. Next, please. Another event is the bloom of, this bloom happened in 2019 in the north part of Oman Sea, and as you can see, 2 tons of marine organisms dead in this bloom event, and duration of this bloom was only 1 day, but with huge mortality. And the causative species was not single species. When we observe under microscope, we found chain form, many destroyed cell, and chain form probably belong to Margarifidinium, and Carlodinium sp was alive, but Margarifidinium was destroyed and destructive. As this research shows, Jeong in 2005 showed that Cochlidinium polycharacoid can feed on Amphidinium cartridge, and we sampled the water the day after bloom, and the next week we found a high concentration of Amphidinium cartridge, and we suggest probably this was Cochlidinium and feed on Amphidinium cartridge, and phylogenetically identified this one and was Amphidinium cartridge, Amphidinium species, morphologically very similar. That's why we use molecular technique and identify this species as a Amphidinium cartridge from this area. Next, please. This slide shows the bloom of Noctulica scintillans. Bloom of these species happen every year after southwest monsoon. And as you can see in this map, it's tropical species and is distributed in all coastal water of tropical from Pacific to Indian Ocean, but was not reported from the Sea of Oman and Persian Gulf before. Next, please. This algae bloom happened in Kish Island in the north part of Persian Gulf in 2021. First, we observed the patch form of this species, Noctulica scintillans, and after few windy days, the bloom spread and covered a large area until January 2020. And this photo have taken by GoPro camera by Etemad Far, pilot Etemad Far. Next, please. Determination the amount of pigments of HAB species during their growth period can give us a good result from the presence of pigment of HAB species in the bloom time, and can be used as a marker. I suggest it's a good marker in the rapid identification of HAB species. In this study, we isolate these species, Gyrodinium instratum and Scripsiella trochoide, from bloom water and bring to lab and provide a unialgae culture and study its life cycle to provide cyst or not, and measure its pigments during the growth period every 5 days. And we had a good result from this. And I think pigments as are a very good chemical marker in the HAB researches. Next, please. I think dinoflagellate cyst mapping in the Rob-Missy area can provide evidence for global spreading or recession of HAB species. Characterization of HAB species in the region phylogenetically describe all HAB species by conventional molecular technique, and it's a good point. To integrate and adopt selective methods for detection, identification, enumeration, and monitoring of HAB species. And I recommend taking international cooperation seriously and carrying out a joint project and integrate technique in the sampling and data analysis. Thanks for your attention.
Thank you very much, Gillian. That was a really nice message to finish on as well. Thank you. All right, so if we can just open up the floor for questions, please go ahead. If you would like to ask Gillian. You can put it in the chat. Oh, yes, please speak.
I don't have the voice.
No, I have a buzzing noise. Does anybody want to ask Gillian the question? Okay, it's obviously very clear, Gillian. Thank you very much.
Okay, thank you.
Thank you.